Identify non-contact approaches such as magnetic fields and dielectrophoresis that provide cell positioning in 3 dimensions without negatively impacting cell viability. Demonstrate in vitro construction of multicellular tissue using 1 or more non-contact cell positioning approaches. Demonstrate survival and functional implantation of a 2 cubic centimeter multicellular skeletal muscle scaffold. Let's construct into an appropriate in vivo model. P2012 plans. Demonstrate formation of vascular elements from endothelial cells within an existing 3-dimensional skeletal muscle. Construct in vitro. Demonstrate directed ingrowth of neurons in an existing 3-dimensional skeletal muscle. Construct in vitro. Title: Nanostructure in Biology 2.843. Description: The Nanostructure in Biology program investigated the nanostructure properties of biological materials to better understand their behavior and accelerate their exploitation for defense applications. This new information about biomolecules unclassified. Volume 1-10. Defense Advanced Research Projects Agency, page 10 of 47, R1, line item number 2. Unclassified. Exhibit R2A RDT&E Project Justification, PB 2012 Defense Advanced Research Projects Agency. Date: February 2011. Appropriation / budget activity R1. Item nomenclature Project 0400 Research, Development, Test and Evaluation, Defense-wide P06011101E, Defense Research BLS1, Bio/Info/Microsciences BA1, Basic Research Sciences B. Accomplishments Planned Programs $in millions, P2010 P2011 P2012, and Complex Cellular Systems provided important new leads for the development of threat countermeasures, biomolecular probes and motors, and neuromorphic sensory systems. This program also developed approaches to mathematically predict a priori the structure of biological materials, especially proteins, based on the desired performance. This enabled the rapid design of new biosensors against previously unknown threats and the design of advanced catalysts based on biological activity to produce new materials of interest to DOD, e.g., tailored explosives. The program also created technology to reliably integrate nanoscale and microsystems payloads on insects that will extract power, control locomotion, and also carry DOD-relevant sensors. P2010 accomplishments. Discovered methods for precise flight control use in combinations of MEMS techniques originating in the previous fiscal year. Developed neural interfaces to insect sensors to complement electronic sensors. Continued development of a protein that preferentially binds to an invariant portion of the influenza virus. Continued design of de novo inhibitory protein of smallpox. Accomplishment / planned program subtotals 34.92853.73939.686 2010 fee, 2011. Congressional Ad, Countermeasures to Combat Protozoan Parasites, 1.600, fee 2010 accomplishments, initiated research to develop countermeasures to combat protozoan parasites. Congressional Ad, subtotals 1.600, see other program funding summary $in millions. N/A. De-acquisition strategy N/A. E-performance metrics. Specific programmatic performance metrics are listed above in the Program Accomplishments and Plans section. Unclassified. Volume 1-11. Defense Advanced Research Projects Agency, page 11 of 47, R1, line item number 2. Unclassified. Exhibit R2A RDT&E Project Justification, PB 2012 Defense Advanced Research Projects Agency. Date: February 2011. Appropriation / budget activity R1. Item nomenclature Project P2012 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 Cost to cost $in millions. P2010 P2011 Base OCO Total P2013 P2014 P2015 P2016 Complete Total Cost CCS2 Math and Computer 38.240 70.001 60.805 minus 60.805 60.670 60.942 67.512 71.512 Continuing Sciences. A mission description and budget item justification. This project supports scientific study and experimentation on new computational models and mechanisms for reasoning and communication in complex, interconnected systems in support of long-term national security requirements. The project is exploring novel means of exploiting computer capabilities, practical, logical and heuristic reasoning by machines, development of enhanced human-to-computer and computer-to-computer interaction technologies, innovative approaches to the composition of software, innovative computer architectures, and new learning mechanisms for systematically upgrading and improving these capabilities. Additionally, this project explores mathematical programs and their potential for defense applications. Promising techniques will transition to both technology development and system-level projects. The accomplishments planned programs in millions. Fee 2010, Fee 2011, Fee 2012 title, Computer Science Study Group, CSSG 6.931 10.550 11.550 description. Current activities include revolutionary advances in 13 topic areas: quantum science and technology, applied biology, biomedical devices and bioinformatics, mathematics, structural materials, functional materials, power and energy, advanced electronics, micro/nano electromechanical systems, MEMS and NEMS, photonics and lasers, manufacturing science and technology, neuroscience, and computational and quantitative social, decision, and behavioral sciences. A key aspect of the EFAB program is DARPA-sponsored military visits. All EFAB principal investigators are expected to participate in one or more military site visit exercise to help them better understand DOD problems/needs. Awarded 33 new grants in the following topic areas: quantum science and technology 4, applied biology 3, biomedical devices and bioinformatics 3, mathematics 2, structural materials 2, functional materials 3, power and energy 3, advanced electronics 3, micro/nano electromechanical systems, MEMS and NEMS 1, photonics and lasers 4, manufacturing science and technology 1, neuroscience 2, and computational and quantitative social, decision, and behavioral sciences 2. Established a mentorship. A mission description and budget item justification. The Defense Research Sciences program element is budgeted in the basic research budget activity because it provides the technical foundation for long-term national security enhancement through the discovery of new phenomena and the exploration of the potential of such phenomena for defense applications. It supports the scientific study and experimentation that is the basis for more advanced knowledge and understanding in information, electronic, mathematical, computer, biological, and material sciences. The Bio and Info/Microsciences project will explore and develop potential technological breakthroughs that exist at the intersection of biology, information technology, and microphysical systems to exploit advances and leverage fundamental discoveries for the development of new technologies, techniques, and systems of interest to the DOD. Programs in this project will draw upon information and physical sciences to discover properties of biological systems that cross multiple biological architectures and functions, from the molecular and genetic level through cellular, tissue, organ, and whole organism levels. Programs in this project also lay the groundwork for advances in military medicine and combat casualty care. The Math and Computer Sciences project supports long-term national security requirements through scientific research and experimentation in new computational models and mechanisms for reasoning and communication in complex, interconnected systems. The project is exploring novel means to exploit computer capabilities, enhance human-to-computer and computer-to-computer interaction technologies, advance innovative computer architectures, and discover new learning mechanisms and innovations in software composition. It is also fostering the computer science academic community to address the DoD's need for innovative computer and information science technologies. Additionally, this project explores the science of mathematics for potential defense applications. The Cyber Sciences Project supports long-term national security requirements through scientific research and experimentation in cybersecurity. Networked computing systems control virtually everything, from power plants and energy distribution, transportation systems, food and water distribution, financial systems, to defense unclassified. Volume 1-1. Defense Advanced Research Projects Agency, page 1 of 47, R1, line item number 2. Unclassified. Exhibit. Protecting the infrastructure on which these systems rely is a national security issue. The Cyber Sciences Project will ensure DoD's cyber capabilities survive adversary attempts to degrade, disrupt, or deny military computing, communications, and networking systems. Basic research in cyber. P2012 increase reflects additional emphasis in basic research for transformative technologies, such as social networking, synthetic biology, dialysis like therapeutics, and quantum devices. The establishment of a new project for cyber sciences. 1. Offset by a reduction for defense efficiencies for contractor staff support and studies. Unclassified. Volume 1-3. Defense Advanced Research Projects Agency, page 3 of 47, R1. Exhibit R2A RDT and E Project Justification, PB 2012 Defense Advanced Research Projects Agency date, February 2011 appropriation, /budget activity R1 item nomenclature project, 0400 research, development, test and evaluation, defense-wide P06011101E, defense research BLS1, bio/info/microsciences BA1, basic research sciences. This project is investigating and developing the intersections of biology, information technology, and microphysical systems to exploit important technological advances and leverage fundamental discoveries for the development of new technologies, techniques, and systems of interest to the DoD. This research is critical to the development of rapid responses to engineered biological warfare agents, radically new biomolecular computers, and novel materials for the DoD. Programs in this project will draw upon the information and physical sciences to discover properties of biological systems that cross multiple scales of biological architecture and function, from the molecular and genetic level through cellular, tissue, organ, and whole organism levels. This project will develop the basic research tools in biology that are unique to the application of biological-based solutions to critical defense problems. The Accomplishments Plan Programs $in millions, fee 2010, fee 2011, fee 2012 title, Biointerfaces 2.000, 2.000, 5.000 description. The Biointerfaces program supports scientific study and experimentation, emphasizing the interfaces between biology and the physical and mathematical computer sciences. This unique interaction will develop new mathematical and experimental tools for understanding biology in a way that will allow its application to a myriad of DoD problems. These tools will help exploit the advances in the complex modeling of physical and biological phenomena. It is also expected that understanding the fundamentals of biology will aid in developing tools to understand complex, nonlinear networks and force structures. This program will also explore the fundamental nature of time in biology and medicine. This will include mapping basic clock circuitry in biological systems from the molecular level up through unique species level activities, with a special emphasis on the applicability to human biology. Operational relevance of this research activity includes improving our understanding of sleep-wake cycles, increasing the scientific understanding of deployment cycle lengths, and enhancing our ability to model the dynamics of disease outbreaks. P2010 accomplishments. Tested theoretical mathematical formulations of developmental laws of biology and demonstrated the existence of fundamental biogenesis pathways that operate across plant and animal kingdoms. Developed novel mathematical tools that decipher complex cardiac signals to detect early warning signs of adverse medical events. Discovered a novel regulatory mechanism controlling cellular protein expression that expands the understanding of biological control systems and how they have evolved. P 2011 Plans. Unclassified Volume 1-4. Defense Advanced Research Projects Agency, page 4 of 47, R1, line item number 2. Unclassified. Exhibit R2A RDT&E Project Justification, PB 2012 Defense Advanced Research Projects Agency, date, February 2011. Demonstrate 2 genomic indicators of geospatial origin of prokaryotic microorganisms. Demonstrate the role of phage bacteria in attack and defense in assigning temporal and geo-localizing data. Demonstrate 4 variable determinations of global origin or residence in a simple mammalian system. Title: Preventing Violent Explosive Neurologic Trauma. Prevent. Medical 4.5003.207. Description: The Preventing Violent Explosive Neurologic Trauma Prevent program seeks to understand the causes of blast. Research will create a model that can be directly correlated to the epidemiology and etiology of injury seen in returning warfighters and attempt to determine the physical and physiological. Tested and validated fabricated device strategies to ensure that they appropriately mitigate the effects of blast brain injury. P 2011 Plan. Investigate the long-term effects of multiple exposures to blast on warfighters following. Exhibit R2A RDT&E. Demonstrate fracture putty in large animal model of bone fracture with independent validation. Initiate expanded large animal studies of fracture putty. Incorporate the identified master antibody molecule into an existing biosensor platform and demonstrate advanced capability in terms of robustness and potential for multiplexing. Unclassified. Volume 1-6. Develop decomposition frameworks and initial cluster of neurobiological mechanisms to better understand their relationship. Develop tools to link analytic frameworks, neural mechanisms, and environmental variables to a particular story. Title: Human-Assisted Neural Devices, Medical 15.97518.250. Unclassified Volume 1-7. Title: Mathematics of the Brain, MOB 1.8726.00010.000. Description: The Mathematics of the Brain, MOB, program will develop a new mathematical paradigm for understanding how to model reasoning. This includes a comprehensive mathematical theory to exploit information in signals at multiple acquisition levels, which would fundamentally generalize compressive sensing for multidimensional sources beyond domains typically used. This program will establish a functional mathematical basis on which to build future advances in cognitive neuroscience. Explore the comprehensive measurement theory's utility in applications such as imaging and radar. Investigate novel forms of prior knowledge in order to improve sparse signal sampling. P 2012 Plans. Develop detailed mathematical prior knowledge representations and associated models for imaging and radar applications. Exploit the new theoretical measurement framework, together with novel forms of prior knowledge in Defense Advanced Research Projects Agency, page 8 of 47 are. These computational models can be used to predict which acoustic signature changes would lead to reduced perceptibility and the brain's ability to learn and adapt to novel acoustic signatures. P 2011 Plans. Develop a detailed theoretical model for manifestly quantum mechanical effects in specific biological systems. Formulate testable predictions for effects of perturbations on the biological system. Experimentally verify that the biological system exploits quantum effect S at room temperature. P 2012 Plans. Develop theory for sensor utilizing biological quantum effects. Design synthetic sensor based upon quantum mechanism employed by biological system S. Model sensor performance. Experimentally probe the limits of biological sensors, exploitation of the quantum effects. Demonstrate initial proof of concept of potential non-electrode-based modalities of neural interface. Identify potential quantitative methods to map structural neuroanatomy and system dynamics for afferent and efferent pathways. Determine whether auditory percepts can be altered with respect to location of a heard object. Investigate how auditory. Unclassified. Exhibit R2. Identify non-contact approaches such as magnetic fields and dielectrophoresis that provide cell positioning in 3 dimensions without negatively impacting cell viability. Identify non-contact approaches such as magnetic fields and dielectrophoresis that provide cell positioning in 3 dimensions without negatively impacting cell viability. Demonstrate in vitro construction of multicellular tissue using one or more non-contact cell positioning approaches. Demonstrate survival and functional implantation of a 2 cubic centimeter multicellular skeletal muscle scaffold. Less construct into an appropriate in vivo model. B2012 plans. Demonstrate formation of vascular elements from endothelial cells within an existing 3-dimensional skeletal muscle construct in vitro. Demonstrate directed ingrowth of neurons in an existing 3-dimensional skeletal muscle construct in vitro. Title: Nanostructure in Biology 2.843. Description: The Nanostructure in Biology program investigated the nanostructure properties of biological materials to better understand their behavior and accelerate their exploitation for defense applications. This new information about biomolecules unclassified. Volume 1-10. Defense Advanced Research Projects Agency, page 10 of 47, R1, line item number 2. Unclassified. Exhibit R2A RDT&D Project Exhibit R2A RDT&D Project Justification, PB 2012. Defense Advanced Research Projects Agency. Date: February 2011. Appropriation. Exhibit R2A RDT&D Project Justification, PB 2012. Defense Advanced Research Projects Agency. Date: and neuromorphic sensory systems. This program also developed approaches to mathematically predict a priori the structure and neuromorphic sensory. Exhibit R2A RDT&D Project